as we saw that RNA sequence data can be generated through various different technologies including Illumina, PacBio or Nanopore. However, once the RNA sequence data has been generated, the raw data has been generated, which comes in what is called a FASTQ file, the next steps are to determine which genes are switched on, how much they are switched on. But before we can do that, there are certain steps that one has to take, and many of these steps we will not go in detail in this course. There would be, maybe in future we'll have more courses which would cover these algorithms in more detail. But there are basically 4 phases of analysis. One is to take the raw reads and align them to the reference genome. So if it is a human data, human RNA seq, you will align all those reads to the human genome. Because these are short read sequences, you will need to assemble them to a gene level or at exon levels. Once you have assembled the genome, assembled all the reads onto the genome, and you have now covered all the exons and the whole gene, you can start quantifying each of the expression values. So you will get a actual count of number of transcripts that were present for a particular gene. Because there are usually a lot of differences amongst various samples, there is a need to normalize across samples, which is a standard for most of the big data that is generated, most of the omic data that is generated. And finally, you model it to figure out differentially expressed genes or transcript. So essentially there are 3 major ways of doing it. One is this pathway A, then is pathway B and pathway C. Pathway A and B have similar alignment protocols, algorithms. However, they differ at the time of quantifying, normalization and modeling, which is different in C with cufflinks and cuffdiff2. Here it's HT-seq and D-seq2 majorly. And then there is a third, another completely new method which is called Callisto-Salmon, and these are the names of the algorithm. As I said, we won't go into details of it, how they are performing and how they are aligning and assembling things. We'll probably cover those in some more details later on. So once you have got these differentially expressed genes, you can, as we saw, you can start building pathways that are differentially expressed among the 2 samples, be it disease and normal, specific processes that are different, and so on and so forth. So you essentially get the functional readout of the genome. How is the genome differing between 2 sample sets? So what we will do quickly, as we did with genomics, we will quickly look at some of the applications of RNA-seq, and this has revolutionized the way we actually understand functions of different cell types, tissue types of a human body. So one of the biggest attempt done to understand the functional differences at transcriptome level of all the tissues across in a human body is called a GTX Consortium, which is genotype expression, right? Now this is a complete atlas of transcriptomes of almost all human tissues. So you can see here there are, we know brain is consisting of several different kinds of neurons and tissues. So all of these are listed here, about 10-15 of them, and then all different kinds of tissues that are present in a human body. Now most of these tissue samples were taken from cadavers, and therefore, because you can't extract certain tissues in a live person, what was done was that once the tissue was taken, the tissue was, the RNA was extracted, what is called bulk RNA extraction were done. That means all the tissue cells that, say for example, if this is from liver, all the all the cells from the liver were taken, RNA sample was extracted, and then they were sequenced to figure out what are the differences that are happening at the local level, which is called cis difference, and what are the differences that are happening at the distant level, right? So, for example, a particular gene can be regulated with the promoter sequence here, which is called cis location because it's close to the gene here, but if there is some long gap, right, and there's a sort of a DNA element which affects the expression of this will be called trans. So there are proteins called transcription factors. They usually act in trans. Anyway, so you can determine both cis and trans differences in expression of various genes and identify what are the different splicing events. So, as we were talking about that exons can be spliced in different manners, these splicing events can also be captured, which could be specific for specific tissue types. So, same gene could have different splice variant in, let's say, liver and in stomach, right? That can happen. So essentially this whole atlas is publicly available for people to understand what are the differences in expression of a, let's say I am interested in gene 1, I can look up the expression of gene 1 across all the tissues in human body, around like 30, 40 odd different tissue types, right? So essentially I can do that for all the 30,000 genes and start building tissue specific gene expression maps. So I will know that what are the genes that are highly expressed in liver, what are the genes that are highly expressed in lung, what are the genes that are highly expressed in kidney, what are the genes that are highly expressed in the cortex of a brain or hypothalamus of a brain, or what are in the stomach or in the bone. So all of this I can do, and this would give. So now if I am looking at, so the utility of this kind of a data set is that if I am now looking at a particular cancer of a particular tissue, I have a baseline. So for example, if I am looking at lung cancer, I know what is the expression level of genes, normal expression level of genes in a lung. I have a baseline. I can now see what happens when cancer happens, lung cancer happens, and I can compare directly. Similarly, I can do with brain tumors, or I can do with liver cancer. So this kind of database, this kind of consortium effort has given us the ability to form what are called the baselines of normal behavior. Any aberration from this can be transcribed to a particular, or can be identified as due to a particular disease, and this something is being continuously used, and this is an example. So for example, you have GTX database, you have different cell types. You can ask within this GTX database that if, because these are from different individuals, the samples have been taken from large number of different individuals. So for example, lung samples have been taken from large number of individuals, and we know that we can also find out that whether each of these patient who cadaver that gave the lung sample had any genetic differences. We can check whether there is a difference in expression of a particular gene based on the genotype, as we saw previously that when the genotype changes, the expression could change. In some tissues you will see a genotype dependent expression difference, in others you will not. So let's say this is brain and this is kidney, right? So particular gene is dependent on the genotype. That means the expression changes as the genotype changes. When you have TT, your expression is low compared to when you have AA in kidney, but that same genotype change doesn't affect the expression of this gene A in brain, and so gene A is only variable in kidney but not in brain, and variability is coming because of the change in the genotype. These kind of interaction analysis can be done, which is very useful in identifying if, say for example, genome-wide association study has been done, we know there is a SNP which is causal, and we know that because of this SNP this kind of expression level difference happens. We can now identify what is the exact function of this particular SNP, and this is something that gives functional readout to GWAS studies. It gives functional readout which are tissue specific. So rather than targeting something for the whole organism, one can be targeting very specifically to very specific tissues. So it becomes lot more focused kind of analysis, and outcomes are more focused on a tissue that is affected rather than the whole body, because if you average across all the tissues, average may not be equal to tissue specific, right? That's the bottom line. So one of the other things that has started happening over the last 5 years is that while transcriptomics was something that is being done currently, and what we said was it was bulk RNA sequencing, which means all the cell types of a given tissue were taken together and RNA was extracted from them. What you can also do is what is called, and this has been happening for last 5 years, and we have a single RNA Seek, single cell RNA Seek atlas available as well, where you can actually, from, let's say, from a liver cell, like if you think of a liver cell would have multiple different kinds of cells. They would have hepatocytes, which are liver cells. They would have different kind of blood cells, because most liver is considered as a organ that purifies blood. Then there would be various other lipid cells, adipocytes, fat cells. So you can actually now take the liver tissue, segregate, divide all these cells, divide that tissue into multiple different kinds of cells, and get expression value of each and every single individual cell. So rather than bulking the sample and getting average effect of each gene expression, we can now start getting each cell level expression, and this is lot more data, lot more higher resolution, which allows you to see, as we will see in next few slides, whether we are targeting, when let's say we are analyzing a disease, which particular cell type is actually causing the difference in for the disease, right? Is it the hepatocyte, which is the liver cell, or is it a blood cell, or is it a fat cell, right? Or if there is a cancer and a treatment is done, are the cancer cells are actually getting affected, or we are just targeting the normal cells? All those things can be now addressed using these kind of technologies. I again won't go into detail of how this RNA-seq is done. Essentially what you do is you try to segregate all the cells based on certain cell segregating techniques, which would involve so isolating them separately and then identifying them through some sort of cell sorter technology, and then collecting them based on that, right? And once you have done that, you would have groups of cells which are phenotypically identical. So for example, you'll have all the muscle cells together, all the normal cells together, all the neurons together, all the blood cells together, like that. And then you can do the sequencing. When you do the sequencing, after you've done the QC and all those things, you will start seeing that gene expression levels change with the cell type. So for example, this is cell 1, cell type 2, cell type 3, this is gene expression, right? Certain cell types have higher gene expression, certain cell types have lower gene expressions. Even at the each individual gene level, there could be no difference between the cell types, but there could be significant difference between the cell types, which would mean that in a tissue, a particular cell has higher expression of a particular gene, whereas in the same tissue, other cell types have lower expression. So now if there was some function that depended on higher expression, you know which particular cell type in which particular tissue will be exclusively affected, not the other one. And there are various visualization tools. One of them is called T-Sneak. Again, we won't go into detail. So essentially what single cell RNA seek allows you to do is to sort of look at the resolution of a single cell within a tissue rather than at a tissue level. So you are sort of diving deeper into a tissue and figuring out differences in the expression of a gene at a single cell level. Similarly, you can do what is called spatial omics, which sort of gives you a method to do gene expression, differential gene expression analysis of regions of a tissue which can be variable. So, for example, in brain you can take tissue sections and do in situ gene expression analysis of single cells, and when you overlay that information you start seeing that there are certain tissues which are differentially expressed for the same gene across the tissue, brain tissue in different parts of the brain, right. So this sort of is called spatial omics, because now there is a spatial information that is also encoded and embedded in the information. In this case there is no spatial information, right, because you have now taken the tissue and split it into multiple different kinds of cells and they are sort of segregated using a cell sorter. Here you are maintaining the relationship of the cells within a tissue. So in a cross section of a tissue the cells remain intact. So for example, one of the papers that came out in 2020, what they did was they wanted to understand what are the genes that are involved in macular degeneration, which is a disease of the eye in which the person ultimately becomes blind and it affects the retina specifically. So what they did, they took the retina of disease in normal patients and did single cell RNA seek, and as you can see in retina there are multiple different kinds of cells. There are rods and cones, right, rods and cones are there, then there are neurons there, then there are basal cells there, right, and various other cells are there. So all these cells were differentially analyzed and they were correlated with the variation that was existing between between disease and normal samples. And what they were able to identify by doing this kind of single cell RNA seek was that they identified two genes, TSPAN1 and TRPM1, which were specifically causal for age-related macular degeneration. And we knew exactly which particular cell type was getting affected, which was these RPE cells. So this means that you not only have the ability to tell what genes are getting affected or what genes are causing the disease, but you also know in which cell their effect is being manifested. So you can be a lot more targeted in doing your treatments. Similarly, if you apply the same technique to cancer biology, which is becoming very common norm these days. So you take a cancer cell, resect, like take out the cancer tissue. Now you have all these cell types, different cell types. You isolate each of the cell types separately using a cell sorter, do single cell RNA sequencing, and then when you now do expression analysis and do T-SNE kind of an analysis, what you see is that the malignant cells are clustering together, non-malignant cell, non-cancer type cells are clustering together. There can be multiple malignant types, there could be multiple non-malignant types. And when, let's say, a treatment is given, you can see whether this particular cell type has reduced in proportion or not, or has been eliminated or not. We don't want these to be eliminated, we only want this to be eliminated. So that gives you a very precise target readout, right? You know exactly what was the outcome of the treatment, because these are the cell types, if they are left after treatment, are going to be the ones that will cause remission. And therefore, it is very important to ensure that these cell type A in this case is eliminated from the patient. So and you can actually use this as a screening tool as well and see which of the cell types, which of the candidate drugs can actually eliminate these specific cell types, not any other, because others are not something that the cells the drug should target. So both as a screening tool as well as a treatment efficacy tool, people are nowadays using single cell RNA seq. Now still an expensive treatment, but yeah. Finally, there is multiomics. So you can do multiomics at a single cell level as well. So as we saw, you have mRNA expression transcriptomics, right, methylation epigenomics, and protein expression, which is proteomics, and then you can also a spatial organization of, as we saw in the brain tissue. So by combining these kind of technologies at either at a single cell level or at the bulk cell level, you can start building a better readout of how different cells, different tissues are affected in a particular disease, or how a treatment actually cures the disease, right. So these are the kind of things that are currently being developed and being generated, like the kind of data both in genomics, transcriptomics, even in proteomics and epigenomics, which is giving us computational biologists a lot of data, a lot of information about how biological systems that seem to be highly interconnected, not only at the cellular level, but even at the tissue level, even at the organismal level. This makes biology very exciting, and this is why big data in biology is one of the most complex set of big data sets available. It's more complex because as we saw during this introductory part of the course, just knowing genomics is not enough. Just knowing transcriptomics is also not enough. And just knowing proteomics is also not enough. You need to integrate all of this. And we also saw that the relationship of the phenotype to the genome, to the transcriptome, to the proteome or epigenome is not linear. Each of these layers of information are not connected directly one-on-one. There is a lot of information buffering, there is a lot of information amplification, and these buffering systems and amplification systems are very context specific. That means in certain cells buffering might happen, but in other cells buffering will not happen. Even in a particular given cell, buffering may happen initially but not at the later stage, or may not happen earlier and happen later. So there is not just a snapshot that we are looking at. Many a time we get to know that we know now that there are certain genes that get activated very early on and then switch off, but their effect, the cascade that they have initiated, remains active for a very long time. So now if you want to capture that particular expression effect, you have to capture it as a temporal or a longitudinal analysis. So these are the sort of kind of questions and challenges and data sets that are available, which makes big data in biology as one of the most exciting fields of big data analytics. And a lot of engineering and mathematical and other tools that are available, algorithms that are available, are now being applied to biological databases, and we are getting insights from those kind of analytics which we would not have gotten if we hadn't applied these. And what we are understanding now about biology would have been impossible to do before big data came into picture. So I hope in the next few classes you will learn more about what are the mathematical and algorithmic basis of doing some of the simple steps of the analysis that we have discussed till now. And in later courses, other courses, we will try to cover few more details of these kind of analysis.